Good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Welcome to the parallel session SMIC 2020. I want to check if the presenter is in this room. Oh, Ms. Luazaini, Mr. Kugan Kumar, Ambe Habadis, Kugan Kumar.
Hello, yes, I'm present.
Okay. Ailia Inshira Hishamudi.
Yes, I'm here.
Okay. Sanmuga Prakasam Selvamani. Is there? Not yet. Jennifer Edwina Eyah Malay. Okay. R. Abdullah Renhard Sulaiman.
Good afternoon, Bu. Abdullah Renhard Sulaiman, presented by me.
Okay. Thank you. Aulia Reski Widyaningrum.
Aulia is also presented by me.
Oh, okay. Thank you. Miss Abdullah Renhard. Mr. Abdullah, sorry. Nurul Muhammad Prakoso.
Present, Bu.
Abdul Basit.
Yes, it's me.
Okay. We have 8 of the 10 presenter. Yes, it's me, the 10 presenter. Yes. Because of some reason. Okay, before we start, we will play you. Yes, someone? We can play the video. We will play the, yeah? Yes, we will play you the opening video. This somewhere. Okay, thank you, Samuel. It is my pleasure to welcome here in this virtual room number 6 for parallel session. My name is Trihandayani Kurniati. I'm your moderator in this afternoon. And before we start, let me explain about the rules in this session. We will play presentation video for each presenter, and then we have 5 minutes to discuss. Before discussion time, please, if you want to ask the question, please type your that you want to ask in the chat room, and I will note it. Okay, and because of the limited time to discussion, please give your question briefly and clearly. Well, we will get started with the first video presentation. This presentation is with the title Screening of Factors Influencing Pululan Production by Basidium melaganum using fractional factorial design by Lo Zaini binti Muhammad Izwan Law. Please, someone.
Assalamualaikum and a very good day to everyone. I am Lozaini binti Muhammad Izwanlo from University Technology Malaysia. The title of my research is Screening of Factors Influencing Pululan Production by Basidium Maranajanam DSM 244 using the fractional factorial design. Firstly is the pullulan. What is pullulan? Pullulan is an exopolysaccharide or polymer produced by the fungus Basidium species. It is a linear maltodextrose repeating unit interconnected by alpha-1,6 linkage. This special characteristic of the pullulan gives it many special application, which is one of it is it can form thin film which is transparent or resistant and impermeable to oxygen. Pullulan has been used in many varieties of application, especially in various industries. So the objective of my research is to develop an industrial medium and cultivation strategy to maximize the pullulan production by Basidium balanogenum DSM 2044 using the fractional factorial design. Next is the methodology. First, I will revive the bacterial culture and maintenance. Next, I will proceed to the selection of the pullulan production medium from previous literature. Then I will do the medium optimization using the fractional factorial design or the plaquette Bowman design. And I will study the growth kinetic of unoptimized and optimized medium using the fractional factorial design. And lastly, I will study it in the batch cultivation in 16-liter bioreactor. For the analysis, I will focus on the cell dry weight and pullulan determination using the ethanol precipitation method. And for the result, the table 1 shows the experimental range and levels of factor using the plaquette Bowman designs. There are 7 factors being studied in this research, which is maltose, yeast extract, ammonium sulfate, peptone, K2PO4, magnesium sulfate, and sodium chloride. And these are the low level and high level in the plaquette Bowman design. And figure 1 shows the Pareto chart for the plaquette Bowman parameter. The parameter that exceed the 2.12 line are the significant factors that influence the pullulan production, which is maltose, peptone, K2PO4, and yeast extract. And table 2 shows the analysis of variance ANOVA on the pullulan production. Based on the p-values, it was identified that maltose, yeast extract, peptone, K2PO4 has given significant effect on pullulan production. P-values less than 0.05 indicates that the factor is significant to the response studied, where confidence level is set at 95%. And coefficient of determination, or R-square, was used to check the fit of the model. The statistical analysis shows that the linear mathematical model is significant with the R-square value of 0.89845. And for the conclusion, the pullulan production from Basidium balanogenum DSM 244 was significantly affected by the maltose, yeast extract, peptone, and K2PO4 from the plaquette Bowman design. And this statistical analysis shows that the linear mathematical model is significant with the coefficient of determination R-square value of 0.8945. And for your information, this experiment is still ongoing and undergo further optimization process. And lastly, I would like to thank for the conference organizer and my bioprocess team in Institute of Bioproduct Development, University Technology Malaysia. Thank you to everyone.
Yeah, thank you. Is anyone to ask to the presenter, please. You can type, or maybe you can also unmute your microphone and directly ask to the presenter. Okay, before everyone have a question, maybe I will start with my question. Lozaini, have you ever worked with bacteria beside this is a mold?
Yes, sorry.
Yeah, have you ever worked with a bacteria to produce pullulan?
No, pullulan actually is specially produced by the fungus. So I focus on fungus.
There's no bacteria have produced pullulan, as you know.
There is from literature, there is some bacteria, but the production of the pullulan from bacteria is very low compared to fungus. That's why we focus on cultivating fungus for this polymer production.
Okay. And when did you get this fungus, this isolate?
No, actually, this fungus strain is purchased from the Germany.
This is not a local isolate.
No.
Okay. Thank you. Anyone to ask? Is there anyone to ask about pullulan? Okay. Thank you. We go to the next video, maybe. Yes, Samuel, please. The presenter number 2, second presenter.
Salam Sujatra and a very good day to everyone. So my presentation is going to be about isolation and identification studies on potential xanthines enzyme producing Trichodoma species from local soil. And I was under supervision of Professor Dr. Hesham Ali El-Sashi. So before I go deep into about my studies, let me give a brief introduction or brief explanation on what is research is mainly about. So the first point is I was assigned to isolate a new strain from the local soil. So when we say the word new strain, it is a must to determine and finalize an optimized medium composition so that we can use that medium composition to enhance the production of any kind of end product from the particular strain. And in order to optimize the medium composition, basically there are two ways to do that. First is which is known as one factor at a time. This is also known as a traditional method of optimizing medium composition. And then the second one is a statistical method where there will be a usage of a software known as response surface methodology. So my project is mainly about comparing by using both of these optimizing method to determine which optimizing method enhance the highest level of end product. And the end product that I was focused on more is xanthines enzyme production. So let's move on to the introduction. So this is how Trichodoma species fungus looks like. So as you can see, the color varies according to the different species. So Trichodoma mainly found on soil. It reproduces sexually, and when it's in initial stage, it will be in white color, and when it reaches towards the final stage, it will be in dark green color. And another main point or another important point that I like to add on here is that instead of using, when we say medium composition, instead of using a synthetic carbon source, I was focused more on using agricultural waste as a sole carbon source, because usage of synthetic carbon source relatively increase the cost of the production medium. So in order to overcome that problem, I was focused more on using agricultural waste as a sole carbon source. So the agricultural waste that I choose is known as corn cob. So this is how corn cob look like. So as I mentioned earlier, using of these corn cobs significantly reduce the cost of production. It is easily available and assimilated, meaning it can be found wherever easily. it is environmental friendly, and the important point is the reason for choosing corn cob in particular is because it consists of highest content of xylene, because xylene is the content that will be degraded in order to produce this xylanase enzyme production. So this is the overall method that involved. First, I started with my sample isolation, followed by morphological characterization, culture preservation, and the step number 4 until step number 9 is all the steps that involve in one factor at a time method, optimization method. So I started out with a different medium composition, followed by different optimization on different concentration level of corn cob. So this is the range. So all this range I finalized according to the previous studies that have been conducted, followed by optimization on different type of nitrogen source, and then concentration of different nitrogen source, concentration of nitrogen source, and then different type of mineral source, and last but not least, different concentration of mineral source. So following that, I moved on with optimizing using the statistical approach, using the response-based methodology. So this is the overall result. So number 1, phylogeny analysis. So according to the phylogenetic tree here, the sample that I was isolated, which you can see here highlighted in yellow color, has a close relation, or I would say as a close distance with Trichodoma asperula. So this is the result for different medium composition. So I've used 6 different type of medium, and medium number 3 shows the highest. So whatever source that available in medium number 3, which started from carbon source, nitrogen source, mineral source, that's where I did my optimization study, meaning I optimized each factor at a time using one factor at a time, as well as the statistical approach. So this is the result for different corn cob concentration. 50 grams shows the highest enzyme production, followed by different nitrogen source. Peptone shows the highest, then different concentration of peptone, where 5 grams per liter shows the highest, then different type of mineral source, where magnesium sulfate shows the highest, followed by different concentration of magnesium sulfate, where 0.5 gram per liter of magnesium sulfate shows the highest. So once I finished complete the one factor at a time optimization method, I moved on with a statistical approach using the response-based methodology. So the main reason for me using this response-based methodology is that whatever source or whatever carbon source that have been optimized that shows the potential or enhance the enzyme production will be further screened using response-based methodology, which means this response-based methodology will help to determine the exact component which enhance and which shows a significant impact towards the final enzyme production. So as you can see here, this is the Pareto chart given by the RSM software. So there will be that it shows 4 sources, which are 4 different sources, which is corn cob, peptone, magnesium sulfate, and yeast extract. So comparing with 4 of these sources, corn cob, peptone, and magnesium sulfate shows the significant impact towards the enzyme production, because whichever source that goes beyond this red line considered shows significant impact towards the final enzyme production. In that case, yeast extract does not included in this in this list because it is beyond, or it is below the red line. So whatever screening process that further will be done, yeast extract will not be included. Then following that, a response surface plot was plotted. So the main reason for using this response surface plot is because we would like to detect the interaction in between these 3 selected components, which means which interaction enhance the better enzyme production. So in this graph, it shows interaction in between these 3 different sources, followed by the response surface plot, the RSM profile. It shows in this RSM profile, the RSM software has set its own parameter. As you can see here, it has mentioned corn cob to be used at 100 gram per liter, peptone 6.47 gram per liter, and magnesium sulfate 0.5 gram per liter. This means using the parameter that has been set by the RSM software, I run my own experimental run using this particular point that has been set by the RSM software. According to the RSM software, the estimated enzyme production is 4.32.70. So in conclusion, this is the overall result. When we compare the optimized media using the OFFET method and optimized media using the statistical method, in comparison of one media which has been optimized using the one factor at a time shows the highest enzyme activity level in comparing with a statistical approach. So from here, what we can be conclude is that the best optimization method to determine whether to determine or to enhance the best optimization method to be used in order to optimize the production medium so that we can enhance the enzyme production is OFFET method, one factor at a time, because it shows the high enzyme activity level in comparing with the statistical method. So that's all from my side, and thank you very much, everyone.
Okay, thank you very much, Mr. Kuga. Wow, very interesting. You get so many information about your isolate. How long you did this research? If I
Hello,
could know.
hi.
Hello, hey, yeah.
Can you hear
How
me?
many? How long? Yes, I hear you.
Okay. So
Yeah.
the overall experiment, it took
Hmm?
around 2 years.
Oh.
And for the isolation itself, because the strain I isolated by myself, so
Hmm.
just for the isolation part, I spent around 2 weeks because
Hmm.
I did a few different trials for the isolation, and
Hmm.
then I went for the OFFET one factor at a time. I spent around 3 months to do, and I have done some repetition as well. And then I did the RSM statistical approach method for another 6 months. So
Hmm.
overall, it took around 1 year and half month, somewhere
Wow.
around there.
That's very good. We have a question from Nurul Muhammad Raposo.
Did you
What
utilize?
method? Yes, what method did you utilize to characterize the isolate specimen besides observing the morphology?
Okay, one of the method I used is by viewing under the microscope. Besides that, I send the sample for the species identification, and then they did the species identification, and then they submitted me the phylogenetic tree. So from the phylogenetic tree, I can give a rough estimation on which species this particular fungus belongs to. But I didn't do any further DNA extraction. So if I do a further DNA extraction, I can get the exact species of the fungus that isolated. So for my case, I just did until up to the phylogenetic tree analysis. So yeah, from there that's a method I use to determine the species, the type of species specimen that I use.
Hmm. Is it clear, Nurul Muhammad, or you still have a question? Please, you can ask directly to the Mr. Kugat. Yeah, Nurul Muhammad, Mr. Nurul Muhammad.
Oh, no, no further question, Bu. Thank you, Mr.
Okay.
Kumar.
Thank you, Mr. Muhammad.
Okay. Thank you, Mr. Kugan, for the interesting presentation, and we can go to the third presenter.
Thank you.
You're welcome.
Assalamualaikum. I'm Alaisha Shmudin. Today I want to present regarding screening of factors that influence cell mass production of Cleveromyces lactis using statistical method. Probiotic, mainly known by the food industry as microbes that give benefit to health of the host through improving the mucosal immune mechanism, that also given term as immunobiotics. Cleveromyces, which known as one of probiotic yeast, named due to the ability to assimilate lactose and convert it into lactic acid as the byproduct. And it usually being used in the lactose intolerance patients, which also being characterized as hypolactasia and alactasia. Hypolactasia means this insufficient of lactase levels, and alactasia means the complete absence of lactase in the gastrointestinal tract that enable them to assimilate the lactose and then make it result the patient to become vomit due to the inability to assimilate lactose. And then Cleveromyces lactis, also known as probiotic yeast, that was chosen as probiotic due to its special ability to improve the digestion of dairy product for the consumption of lactose intolerance patients. Okay, this study was made to give the high amount of cell production of Cleveromyces lactis with the selected media and selected condition. Okay, first, I did the working cell bank preparation, and then media cultivation selection, and then bioprocess and medium optimization by RSM. For working cell bank preparation, I did the preparation from the cell itself, and then I cultivate it in shaker rotation in 200 RPM and 30 degrees Celsius. Okay, for media cultivation selection, there are 7 medias that are selected in this research. Based on previous research, there are 7 medias that using a similar strain of Cleveromyces lactis, and then I used the media that contained in previous research, and after that I selected and optimized it by using plaquette Berman. Okay, these are the results. First, for the first cultivation, YPD media, which composed of yeast extract, peptone, glucose, and agar, I made as control. And then I also make another 7 medias that contain different carbon and nitrogen sources, and also another minerals. And finally, it's shown that the most suitable media that give the high impact toward the production of Cleveromyces lactis is media number 7, which composed of glucose, malt extract, yeast extract, and peptone, which being incubated in the condition of 200 RPM and 30 degrees Celsius. This media also was chosen based on the similarity of the type of strain that I've been using in this research, which is NRRLY 8279. Okay, and then after the media cultivation selection, I used the Beckett-Berman RSM method to know what is the most significant media that contain in that medium, give the impact toward the production of Levoromyces lactis. And based on the graph shown in figure 1, it shows that all the factors that being examined based on media number 7 are significant. And the most significant, which give the high impact, is glucose, followed by malt extract, peptone, and lastly yeast extract, which all these four medium contain carbon and nitrogen sources. Okay, for the conclusion, Levoromyces lactis, the most suitable condition is incubated on 30 degrees Celsius for 24 hours in every cultivation. The most suitable media for optimization of lactis is media number 7, which composed of glucose, yeast extract, malt extract, and peptone. And the selected media that being optimized using RSM found that all four materials are significant for growth of lactis. Okay, that's all from me. Thank you and Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Miss Ailia. Yes. Does anyone have a question for Miss Ailia? Please. You can directly ask. Okay. I also work with probiotic, but I use bacteria from fermented food. This is interesting for me. I want to know where this isolate come from.
This bacteria I got from USA that already being isolated.
Okay. So is this isolate been selected for probiotic characteristics such as acid or tolerance to bile salt, etc.?
Sorry?
You confirm this characteristic?
Actually this is not probiotic bacteria. It is probiotic yeast.
Yeah, but for probiotic, there is a characteristic that they must have, such as tolerance to the biosalt or acid.
The research is still ongoing, and I will test it. The what we call it, acidity and the acidity inside our gastrointestinal.
Yeah. Before it can be used as a probiotic. Commercial probiotic, I mean.
Yeah. I will test it.
Okay. This is interesting. Okay, thank you, Ms. Ailia.
Yeah. Okay, thank you.
And we can go to the next presenter. With the title Improvement of Biomass Production by Lactobacillus. Please, Samuel.
Greetings everyone. So my name is Shanu Pragasham. I'm a PhD student from Institute of Bioproduct Development, University of Technology Malaysia from Malaysia. So today I would like to present about improvement of biomass production by Lactobacillus rhamnosus using double carbon source cultivation strategy. So without delay, I continue with my slides. Okay, so first of all, I would like to highlight upon one bacteria we call Helicobacter pylori. It's a common gastrointestinal bacterium which found in human gut, and it is also a causal agent for the gastric ulcer. Approximately 50% of the world population has been recognized has been infected with this bacteria, which can lead to gastric ulcer and severe impacts will be gastric cancer. So this bacteria is currently become very serious infection since it have various antimicrobial, antibacterial properties, resistant properties. So therefore, we are looking for alternative treatments, especially like probiotics. So among various probiotics that's available in market, we are proposing Lactobacillus reutree because this Lactobacillus reutree have various impacts on the treatment against Helicobacter pylori. First of all, if you see Lactobacillus reutree, they are able to produce various bioactive compounds such as reutrecycline, reutrin, bacteriocins, and inhibitory proteins, which can have a direct inhibitory effects on the Helicobacter pylori. This has been scientifically proven. And then, secondly, it also will reduce the effect of antibiotics and other medication that has been used for the patients. Helicobacter pylori, they use various antibiotics at the higher dose. So these antibiotics, what will happen at the end, it will kill off the natural microflora that found in the human gastrointestinal system. Therefore, if we use the Lactobacillus reutree supplements for the eradication of the Helicobacter pylori, so this Lactobacillus reutree not only can kill the Helicobacter pylori, but it also can reduce the effect of antibiotic if we need to use. And thirdly, it also will restore the normal microflora. So it will restore the healthy gastrointestinal microbiome again after the infection during the recovery process. So this will lead to a balanced gut microbiota regrowth again. So therefore, we are proposing Lactobacillus reutree as a potential probiotic strain for Helicobacter pylori treatment. In various Western countries, it has been widely used for the Helicobacter treatment. However, when we looking at probiotic treatment, so the effective dosage for the probiotic treatment to functioning will be at least 10 to the power of 7 CFU per gram. So why it should be 10 to the power of 7 CFU per gram? Because of the condition that it has to pass through. Stomach is acidic, where your duodenum will be alkaline and more to neutral and everything. So when it's go through one stage by one stage, the number will be decreased. So therefore, it must be produced in large amount, higher cell density. It is the only solution to increase the effectiveness of the probiotic treatment. So in our study, we focusing on high cell density cultivation of Lactobacillus reutree. So our strategy that we use is Lactobacillus reutree displays a heterofermentative metabolic pathway where it's not like other bacteria, it can have two carbon metabolism. It have two carbon metabolism where phosphoketalose pathway and also Embedden-Mayerhof pathway. So in order to enhance the cell mass production, we try to use double carbons that can be metabolized by two pathways together. Lactobacillus reutree is so unique that it can use both pathways together compared to other probiotic microorganisms. It has been proven also scientifically. Therefore, we use this strategy in our study where after we do cultivations, recovery of the strain from the cholesterol stock, we try to do optimization by statistical methods. We use two approaches. One is plaquette Burman design to identify the significant components in the MRS medium, commercial MRS medium, and then we try to introduce double carbon source there so that we will identify whether this double carbon source could be the significant or not. And then we try to, after identifying the significant, then we optimize the concentration by using box Benken design. So basically it's a response surface meteorology design, RSM. So we have shown here our low and high values. Therefore, our result here, if we see, okay, number one, based on the plaquette Burman design, we have identified lactose and glucose, both double carbon sources, play important role, a very significant role in the biomass productions. This is what is shown by the Pareto chart here. And then, of course, the nitrogen source comes on the third place, where the yeast extracts also being playing important role in the biomass production. So these are the three components which is significant players on the biomass production of the Lactobacillus rhamnosus, which we're going to optimize on the RSM method. And this analysis by using blanket Berman design is significant by proving by the R squared, where the value is 0.8519, where this model is accepted significantly. Okay, then we move on the RSM. So when we run the RSM optimizations, we try to optimize only the significant components. Rest of the components we cannot omit because Lactobacillus naturally is a complex medium requirement. Therefore, other components we maintain as the previous one. So only these components are consideration we are optimized based on previous slide. So based on our result here, so this model has been proposed is significantly also valued because the F value is 69.89 and then the adjusted R square is 0.9337, where this means about 93% of the variations response could be explained by the derived second order polynomial equation which has been predicted in this study. Therefore, this optimization process is valid. Therefore, we try to compare the unoptimized commercial MRS medium which is using only one sole carbon source, and then our medium which have double carbon source, which in the suggested concentration 30 gram of lactose, 45 gram of glucose, and 33 gram of yeast extract could induce fourfold of biomass produced from the normal commercial MRS medium. So here we have predicted that 4.35 gram per liter of cell dry weight we can obtain from this formula, and we managed to get the actual reading was 4.12 with the triplicate runs of the experiment. So this proves this formula is valid, and then we also able to produce 4 times higher the biomass compared to commercial MRS medium, which is using sole carbon source, which is only glucose 20 gram per liter. So the double carbon source containing modified MRS medium, which is proposed, is based on the nature of the bacteria, where it is sit in gastrointestinal environment and it is always encounter multiple carbon sources in the human gut. Therefore, we use this nature to cultivate in the laboratory to speed up the cell growth, and this cell growth increase the higher cell mass productions. Therefore, this study is funded by the University Malaysia, University Technology Malaysia's ICOA grant, and we would like to thank the university for the support for the research work. And these are my references, and thank you.
Okay, thank you. For Mr. How can I call you? Your name is
Hi.
so long. Yes?
Just call me Shan. Shan.
Shan.
S-H-A-N. Shan.
Shan. Okay, thank you for the presentation. And okay, participant, is there any question to Mr. Shan? I think Mr. Shan and Miss Ailia and Mr. Kugan, Miss Luazaini, come from one lab?
Yes, from one lab. Supervised by Prof. Hisham.
Yeah, I know him. Okay, but okay. Any question? No? Well, I want to ask you, as we know that the Helicobacter pylori was difficult to inhibit because it has resistance to gastric acid. Yeah, and so it can grow in stomach. Is this your cultivation strategy? Could increase the capability of this Lactobacillus rhamnosus to inhibit the Helicobacter pylori?
Yes, because it has been proven scientifically,
Mm-hmm.
even though it's found on the, actually it's not found on the stomach, it's found in the ileum, lower abdomen, but
Yeah.
they prove that it produced some kind of bioactive compounds,
Mm-hmm.
and it's also well known because of the name Ru3. It produces a kind of antimicrobial compound which is widely used for the food industry nowadays. So this Ru3 compound also have specific effects on the Helicobacter pylori. So that's why from the ileum, but God created it should be seated in the ileum. But
Mm-hmm.
we found medically and we proven scientifically that we can, if we cultivate and we consume this probiotic bacteria, it also can treat the stomach problems, especially the Helicobacter. So in Western country, in the hospital itself, they give these medications. So that's why we're proposing how to cultivate. The problem of probiotic cultivation is they produce lactic acid, and this lactic acid will inhibit the biomass. So usually all the probiotics products, they will have less CFU. So that's why the world keep on doing research how to increase this biomass. So my study is to increase the biomass industrially. So our strategy is look for the double carbon. That's the thing.
And this medium that you use could inhibit or eliminate the effect of the acid
Actually, that
that
is
the
not
bacteria produce?
not part of my study actually.
Okay.
But my study is more to biomass. But me and my supervisor, Prof. Isham, we are discussing after we get this biomass, high cell density, we will try to test also. But that is another part, maybe apart from
Okay.
my work actually.
And once again, it has to the reutrin, retrocycline, and bacteriocin. You mentioned it in your presentation.
Yes.
What the difference about these three components?
Okay. So the first one, deuterin, is a secondary metabolite product. So it's not a peptide. It's a secondary metabolite product. So it is
This is not
produced from the
antibiotic.
it,
Sorry.
pardon?
This is not antibiotic. Bacteriocin is not
Yes.
antibiotic, but reutrin is antibiotic or?
Deuterin is also antibiotic.
Okay.
Retrocycline is a peptide. Retrocycline is a peptide. Peptide produced by the bacteria. And then now we are testing another one, which is a bacteriocin. And then this bacteriocin is more like antibiotic compounds, like we have the streptomycin, something like that. So this bacteriocin, so far no paper to prove to name this one, not yet. But they already proved there is a there is a gene code, because our strain is not isolated. Our strain is from the DSM, and then there is
Commercial
other.
strain.
Yes, it's a commercial strain.
Okay.
It is
Yes.
isolated from infant gut, so the baby's gut. So many people working on it, and latest paper prove this strain got the gene to produce bacteriocin. So it can produce bacteriocin, totally different. We don't know the nature. And second thing, it can produce a retrocycline, antibiotic peptide. It's a peptide protein. And then the third one, if we give glycerol, it can convert the glycerol into reutrin. So reutrin is a third compound. So got three microbial compound that can be produced by this one.
From one isolate.
Yes, from one isolate. That's a unique for this reutrin.
Yeah. So you use a cultivation strategy to make these three optimal. These three.
Yes. We are checking in our optimized medium, we are checking presence of these three. But just the bacteriocin, since we haven't identified, so we are facing some difficulties on that. But others we are identifying now actually.
Okay, thank you very much for your explanation. Okay, we go to the next bioprocess optimization. Sorry, for high biomass production of Lactobacillus casei. Yeah, please, Samuel. Still loading. Okay. This one. Okay. Is it not clear? Still not clear?
And hello everyone, and this is my presentation title for today, which is the medium optimization for high biomass production of lactobacillus casei in shake plus level. And I am Jennifer, and I am from School of Chemical and Energy Engineering, University Technology Malaysia, and Institute of Bioproduct Development, University Technology Malaysia. So for the introduction, as we all know, consumers are giving more importance to the food choices they make, and they tend to choose the food product which not only satisfy their hunger but also improves their health and well-being as well. So this preference creates a niche for functional food among the conventional food market. Probiotics is one of the blooming clusters of functional food, and it is the biggest functional food product in the world. So for the methods, firstly, we have to establish the working cell bank for lactobacillus casei bacteria from glycerol stock, which is obtained from the Institute of Bioproduct Development. And then a literature review was carried out to choose suitable medium for lactobacillus casei cultivation. And out of the literature review, 6 medium was selected, and those 6 medium was selected for further screening using OFAT approach, which is one factor at a time approach. And then the optimization study was carried out using the shaker incubator at 200 RPM, 37 degrees Celsius and 48 degrees Celsius and 48 hours to obtain the biomass production. And cells are activated on MRS agar medium and MRS prot for inoculum preparation, and 250 shake flask containing 50 ml media at 200 RPM, 37 degrees Celsius and 24 hours. The initial pH was adjusted at pH 6.2. The cell biomass was measured using the optical lens using the spectrophotometer. And for the results and discussion, firstly, this is the results and discussion for the medium screening from the literature review. 6 mediums was chosen for the medium screening using OFAT approach, and medium number 3 was selected as it supports the higher cell mass and lower lactic acid production. And the medium number 3 contains sugarcane molasses, peptide, yeast extract, magnesium sulfate. And this produced the highest cell biomass, which was 2.74 gram per liter with pH at 3.73. This simple medium is a simple medium as it only contains four components, and it produces high cell biomass, and this creates an opportunity to reduce the cost, energy and time involved in the bioprocessing area, and thus this is an advantage to use a medium like this as it contains 4 medium components only. So this medium was selected to undergo the further screening using the OFAT approach. And as an overall result, this is the comparison between unoptimized and optimized medium. So as we can see, the OFAT optimized medium produced 6.9 gram per liter of cell mass compared to the unoptimized medium, which produces only 2.46 gram per liter, and this is a 2.68 fold increment in the cell biomass. The OFAT optimized medium contained lactose, soybean meal, yeast extract and magnesium sulfate. As an overall conclusion, this study has demonstrated that the OFAT method for optimization of medium components has created a higher cell mass compared to the unoptimized medium, and it has produced 2.8-fold improvement compared to the unoptimized medium. So initially, different carbon and nitrogen sources were tested for their contribution for the cell mass improvement, and out of them, lactose and soybean meal had shown the more positive result for the biomass production. At the end of the experiment, medium, which consists only four components, lactose, soybean meal, yeast extract, and magnesium sulfate, has been developed to produce high cell mass of 6.9 gram per liter. And then the optimized medium composition developed from this experiment has the potential to reduce the overall cost of the medium and provides important findings for the large-scale cultivation of lactobacillus casei biomass production. That's all for my presentation today. Thank you.
Okay, thank you. Okay. Apologize to Mr. Nur Muhammad. Your question? Okay. To Mr. Shan. Yeah. As usual, if anyone have a question for Mrs. Sorry, yes. You talk about lactobacillus casei? Yes. Yeah. Anyone have question? I'm still waiting. Yeah. No one? Okay. What I call you, Miss Gianna? Yes.
Jennifer.
Oh, sorry. Jennifer. Miss Jennifer. Yeah, you work with Lactobacillus casei, and you find the best medium for
Yes.
biomass production.
Yes.
Yeah, and for conversion, you have the medium. The best medium is the medium that contain soybean meal, not
Yes.
molasses.
No. Soy molasses was used as a carbon source, and then during the carbon source optimization, different carbon sources were used, and among them, lactose performed better than molasses. So lactose was chosen to proceed the experiment, while
Okay.
soybean was used as a nitrogen source. And among the different type of nitrogen sources used, soybean meal performed better for biomass production.
Because this is the lactic acid bacteria, so this bacteria prefer lactose than
Yes.
glucose or sucrose in molasses. Yeah.
Yes.
What's the next research that you want to provide?
After this, after following this OFAT optimization, this will be followed by statistical optimization. And after
Oh.
statistical optimization, the cultivation will be carried out in larger scale in bioreactor.
Okay. This is also the commercial strain
Yes, this
or
is also,
local strain. Oh,
this is commercial strain.
commercial strain. Okay. Yeah. Thank you. Anyone have question? Even you are in the same lab, you can ask to your friend, Mr. Shan. Maybe, or you also have the answer because you work together in the same lab. Okay. We go to the next presenter. Thank you for
Thank
Miss.
you. Thank you.
Next presenter, please, Samuel.
Good morning. My name is Nur Muhammad Prakoso. I am representing the Human Genetic Research Center in FLE. I will be presenting our research on variant identification in exon 11 of galactosamine and acetyl 6 sulfatase or downstream in mucopolysaccharidosis type 4A patients in Indonesia. First, let's talk about the mucopolysaccharidosis. Mucopolysaccharidosis is a group of lysosomal storage disorder which is caused by deficiency of lysosomal enzyme that leads to the accumulation of glycosaminoglycan in the lysosome. In case for MPS4A, the patients are experiencing deficiency of Down's enzyme, which in turn leads to the abnormal storage of creatine sulfate and chondroitin 6 sulfate in their cells. According to previous research, MPS4A is caused by pathogenic mutations in the GANS gene. The translation product of faulty gene leads to the malfunction of GANS enzyme activity. The inability of the cell to degrade creatine sulfate and chondroitin sulfate initiates the cell to activate the cell death pathway, which over time will be progresses into MPS4A phenotypes. Recent genetic studies in exon 11 of GANS gene successfully reported numbers of pathogenic variations in MPS4A patients. However, genetic analysis of MPS4A patients in Indonesia has not been conducted before, and so this study focuses to exon 11 of GANS gene in MPS4A patients in Indonesia because several pathogenic mutations has been identified before in this action in another MPS4A patient population. Let's move on to the method. 7 MPS4 patients and 30 normal individuals were agreed to be involved in this study. This study is initiated by DNA isolation, primer design, polymerase chain reaction, DNA sequencing using Sanger method, and then finished by sequencing analysis. Move on to this result and discussion. The visualization of PCR product shows that a specific MT count with 331 base pair in size had successfully amplified. Furthermore, the sequencing results shows here showing clear peaks and high quality peaks without noises, which can sometimes interfere with the sequencing reading. All the sequencing results of patients and normal individuals are aligned here with reference sequence acquired from NCBI. We identified a substitution from guanine to thymine at coding position number 1177 and alters the 393rd amino acid from alanine to serine, which can be observed here in one patient and 5 individuals. However, this mutation has been identified before and is still in number of databases. Although reported, we perform pathogenicity analysis using in silico tools, and finally we classified this variant as benign, and our analysis results is consistent with the previous report that described this variant as benign. To finish the presentation, this study provides novel data in exon 11 of GANS gene and MPS4 patients in Indonesia. A variant which has been identified in exon 11 of GANS gene in Indonesian population is alteration of the 393rd amino acid from alanine from serine. This variant has been previously reported as benign, and then we wish to perform another research to characterize another variants in another exons of the GANS gene in Indonesian patients in mucopolysaccharidosis. Thank you.
Okay, thank you, Mr. Nurul Muhammad. Yeah, any question, everyone? Yeah. Yeah, please ask the question. No one have question? Again, this is research come from different field, yeah. the first we talk about microbiology, and this is a molecular in the medical.
Yes,
Yeah.
Mystery.
Okay. Well, I want to know that according to your research, this mutation, yeah, you found the mutation.
Yes, we found an alteration of a single base substitution
Mm-hmm.
in the in normal individual.
Mm-hmm.
I'm sorry, in a patient and normal individual.
But this mutation has found before in Japan.
Yes, correct, and has been classified as benign or not causing
Mm-hmm.
the disease.
How about the Indonesian patient?
Um, the
You
mutation is
get
of
the blood from the patient, or you get the blood to extract your the DNA from
yes,
the
correct.
patient.
Yes,
Okay.
correct. We collect the blood from the patient and then
Mm-hmm.
we extracted the DNA, perform amplification of the segment of DNA, and then we perform sequencing to detect if there is any variation present in the patient.
So this mutation does not contribute to the uh, what it cause to the patient,
Yes, correct.
the
But
disease, the disease.
yes, correct. But
Mm-hmm.
if we find the same mutation
Mm-hmm.
in another patient in the future, we
Mm-hmm.
can determine that mutation is not the cause of the disease.
How many sample you get from the patient?
In this research, we use 7 patient sample and
Mm-hmm.
30 normal individuals as control.
Mm-hmm. And it show the different from the control.
The mutation is also
Mm-hmm.
present and in control.
Oh,
So it's
okay.
more likely to be polymorphism.
Okay. Gans enzyme. What is the function of this enzyme?
Based on the previous research, this enzyme is responsible to cleave sulfate compound from creatine sulfide and heparan sulfide. So
Mm-hmm.
abnormality of the activity of this enzyme leads to the accumulation of specific compounds in the patient cell.
Oh, and but there's no effect to the cell, to the metabolism of cell.
Of course, there is an effect.
Mm-hmm.
The accumulation of specific compounds leads to the cellular stress.
Mm-hmm.
So the cell initiates the cell death pathway.
Mm-hmm. Yeah. Okay. Thank you, Mr. Nurul Abdullah. Yeah. Okay. Anyone? Anyone have question beside me? No. Okay. We can go to next presenter.
Good morning. My name is Nurul Muhammad Prakoso. I am representing the Human Genetic Research Center in FKUI. I will be presenting our next research on the identification of novel mutations in exon 1 of iduronate 2 sulfatase or IDS gene from mucopolysaccharidosis type 2 patients in Indonesia. First, I'm going to introduce about the disease. Mucopolysaccharidosis type 2 is an X-linked recessive lysosomal storage disorder which affected by the presence of pathogenic mutations of IDS gene, which causes abnormal storage of dermatan sulfate and heparan sulfate because the faulty gene produces enzyme with damaged function. This condition initiates the cells to activate cell death pathway, which the damage will affect the tissue and organs. This condition is progressive over time until the person with faulty gene develop MPS II symptoms. Iduronate 2 sulfatase or IDS is responsible for the synthesis of IDS enzyme. The size of IDS gene is 28 kilobases and located on the longer arm of chromosome X and expresses 550 amino acids to synthesize IDS enzyme. In this study, the exon 1 of IDS gene is observed because the role of exon 1 in IDS mRNA signaling to endoplasmic reticulum. The sequence of exon 1 is translated into 33 residues, which is consisted of signal peptide and propeptide. As can be observed here, the first 25 residues are formed to be signal peptide, which will be identified by a protein RNA complex called signal recognition particle or SRP in cytosol. Therefore, it is important to ensure that a sequence of exon 1 doesn't have any alteration which will affect the function of signal peptide for translocation of mRNA IDS ribosome complex to endoplasmic reticulum. Let's move on to the method. 10 MPS II patients and 50 normal individuals were agreed to participate in this study. This study is initiated by DNA isolation, primer design, polymerase chain reaction, DNA sequencing using Sanger method, and finished by sequencing analysis. And then results and discussion. Based on the amplification result of patient samples, shows a specific band with 483 bases in pair, 3 bases pair in length, as seen here, the sequencing result also shows clear and high quality peaks without interfering noises in the electropherograms. And then the sequence alignment was performed by aligning the consensus coding sequence or CCDS of IDS gene and the exon 1 of sequence from the samples. Based on the alignment, 2 mutations found in P9 in coding number 22 and coding number 54. The first mutation, the alteration of cytosine into adenine at coding number 22, is a silent mutation which changes the codon from CGA into AGA, and both of them are translated as arginine. So no alteration of amino acid take place. The second mutation, the alteration of cytosine into adenine at coding number 54, is a missense which changes the codon AGC or serine into AGA or arginine. Based on the American Medical Genetics and Genomics guidelines, a mutation that doesn't affect the gene product is classified as a benign mutation. A silent mutation shown in this study at coding number 22 doesn't result in any structural and functional changes of IDS enzyme. And the second mutation, at coding 54, we conclude that this mutation is benign since we predict that it doesn't cause potential harm to the structure of gene product. Both mutations are not located in the peptidase cleavage sites to be processed in the reticulum endoplasmic. In theory, the mutation is considered to disturb the cleavage process when the mutation causes the signal recognition particle, the SRP itself, unable to identify amino acid in the cleavage site due to amino acid substitution like this one. But in case for IDS enzyme, the important amino acid is located between codon 23 and codon 24, and between codon 25 and codon 26. So this mutation located at codon 8 and at codon 18 doesn't located at the important amino acid. To remark this presentation, this study has successfully detected 2 variants in exon 1, which are predicted not to cause any structural changes on the IDS enzyme. We also hope to conduct another experiment to find variation in exon 1 of more MPS II patients in Indonesia. Thank you.
Okay, thank you. I'm very sorry. The previous presenter was Mr. Abdullah. Yeah, and this presenter was Mr. Nurul.
Yes, correct, Miss Tri.
Okay.
Make
Yeah.
make a effort for the develop this skill. So what we doing?
Yes. Okay. Anyone want to ask? Giving the question for Mr. Nurul? Yeah, this is still about mucopolysaccharidosis. Yeah,
Yes, correct.
what kind of disease it is?
Mucopolysaccharidosis type 4A and type 2, the previous presentation, is actually located in the same group of disease called mucopolysaccharidosis. But the difference between mucopolysaccharidosis type 4A and type 2 is the gene responsible for making the enzyme, and the enzyme which are responsible for degrading certain compounds.
And if someone have this disease, what I mean? I'm sorry. Sorry. This is some error in my. Okay. Okay. I have technical error. I'm sorry. Yeah. What kind of disease? If someone have a mucopolysaccharidosis, what type of disease they have?
They actually group as lysosomal storage disease. So the abnormality is located in the degradation of several compounds in their lysosome, in their cells. So it depends on the accumulation of certain compounds in different organs. In case for mucopolysaccharidosis, the compounds is lots can be found in a high number in skeletal. So in patient with mucopolysaccharidosis,
what symptoms?
Mostly they represent symptoms such as skeletal abnormality, skeletal dysplasia. So the main symptom is the lack of growth.
Okay. It affect to the bone, maybe.
Yes, the bone cells have abnormality.
Abnormality. Okay.
Yes, and they tend to die. So
Okay.
so growth cannot take place.
And this is a genetic disorder.
Yes, correct.
Okay. In this research, you found 2 mutations in one gene.
Yes, we found mutation in 2 mutation in only in exon 1.
Okay. But no significant effect.
We predict that this mutation have no significant effect on gene product.
On gene product, the product is enzyme.
Yes, correct.
Piduronate-2 sulfatase (IDS) enzyme.
Yes, correct.
Deficiency to this enzyme. Okay, thank you for the explanation for Mr. Nurul. Yeah. Next, we can finish this early, maybe because no one asked a question. Okay. Next presenter.
Good morning. My name is Nurul Muhammad Prakoso from the Human Genetic Research Center in area FKUI. I will be presenting our research on role-based alterations at intron 3 of 6-pyrophile tetrahydroxyrin synthase gene in Indonesian patient population. First, let's take an insight into the disease. Tetrahydrobiopterin or BH4 deficiency is a disorder which is caused by 6-pyrophile tetrahydroxyrin synthase enzyme deficiency. This enzyme is required in the biosynthesis of tetrahydrobiopterin, which is an important compound in the metabolism of phenylalanine. Persons with deficient PTPase enzyme are unable to produce the required tetrahydrobiopterin, which in turn leads to the accumulation of phenylalanine, or in clinical background called hyperphenylalaninemia or HPA. The incidence of patients with deficient BH4 can be found in 1 of 500,000 to 1 million newborns globally, which is extremely rare. The deficient level or function of PTPase enzyme is linked to the presence of pathogenic variants in the PTPase gene. The gene is consisted by 6 exons and 5 introns. However, in this study, we analyzed intron 3 because pathogenic mutations, which is called aberrant splicing events, has been detected before. This research is also the first molecular genetic study performed in PTPase patients in Indonesia. Let's move on to the method. 1 PTPase patients and 33 normal individuals were agreed to be involved in the study. The study is initiated by DNA isolation, primer design, polymerase chain reaction, DNA sequencing using Sanger method, and finished by sequencing analysis. The visualization of PCR product shows that a specific amplicon with 645 base pair in size had successfully amplified. However, noises are detected in the sequencing result, but the main reads can still be distinguished from the noises. All the sequencing results from patients and normal individuals were aligned with reference sequence acquired from the NCBI. And based on the sequencing result, the alignment result, we have detected 29 variants, as can be seen here, which is consisted by 23 substitutions and 6 insertions in the sequence of intron 3 and 4. In theory, the presence of variants in splicing regions between intron and exon may disturb the recognition of splicing site by the spliceosome. For example, an intronic variant that were published by Liu et al. Found that the alteration of guanine from guanine to adenine at the +1 donor site cause exon 3 skipping event that leads to the production of truncated protein. However, in this study, we identified more than 5 variants which is located in the splicing site or close to it. However, we could not characterize their functional effect because whether they cause disturbance to splicing process or not. Therefore, we cannot classify the variants into pathogenic or benign variant. To finish the presentation, this study has found 29 variants in intron 3 and 4 of PTPase gene. We hope to perform a functional analysis using an RNA sample to ensure the effect of the variants. Thank you.
Okay. Thank you, Mr. Nurul. Yeah, I'm still waiting for the question. Mr. Kugan, maybe? Miss Alia? Miss Jennifer? Anyone? Anyone? Mr. Shan? Is still there? Mr. Shan? Okay. Yeah. I will ask as the previous presenter, I want to ask about the hyperphenylalaninemia. Can you explain what this is about?
Well, thank you, Mr. Sri. It's me again. So the
Yeah.
disease is a condition where the deficiency of certain types of enzyme, which causes the accumulation of phenylalanine in the cells. There are three enzymes which actually can be responsible in the increased level of phenylalanine. There are guanosine triphosphate or GTP, ETPS, and SR protein or SR enzyme. But in Indonesia, we suspect that one patient in Indonesia, carries mutation in PTPS patient. In PTS gene. Sorry. So we perform genetic analysis in PTS gene to discover whether any disease-causing mutation in the patient.
No information about mutation in other enzyme for Indonesian patient.
For now, there are no information about the mutation in another gene, because the nature of genetic disease is extremely rare. So we are in this research, we only involve one patient.
Okay. What symptom he or she have? The patient, I mean.
Where the accumulation of phenylalanine can be systemic. So the symptoms can be found in several organs and a tissue, and can be detected by clinical examination.
If you want to do the research for the next research, what do you want to do?
Yes, we are delighted to perform next research to characterize the effect of this variant, because we detect lots of mutation which are located in splicing site that can potentially alter the splicing events, whether they are alternative splicing or the abnormal splicing, which can lead to the production of abnormal gene product.
Okay, to the protein, yeah. Protein from translation, I mean.
I think we can use mRNA.
Oh, okay.
Yes.
In transcription.
Yes, correct. To see whether the splicing is changed or not.
Okay. It is difficult to do this research for you.
It's actually quite difficult, because in several steps, such as design primer and sequencing, we have to send the samples to another country. So it takes time to perform the research.
Oh, you design the primer?
It's actually my group research. It's actually my group research. So the research is performed by students from the Department of Biology in UI. So here I try to present the result.
Okay. Yeah, thank you, Mr. Panarul.
Yes, thank you, Ms. Tri.
Success. And then the next presenter, Mr. Abdul Basit. This is the...
Assalamualaikum warahmatullahi wabarakatuh. My name is Abdul Basit. I'm the doctoral student of Biology Department of the Faculty Mathematics and Natural Science Universitas Indonesia. I will be presentation about the species inventories and conservation status of groupers from family Serranidae from Madura Island, East Java, Indonesia. The author of this research is me, Abdul Basit, and then Mr. Abinawanto and Mr. Yasman from the Department of Biology Universitas Indonesia, and then Mrs. Eni Kusrini from the Research and Development Institute for Ornamental Fish Culture. Not to mention, The grouper fish is the primary marine commodity around Madura Island, East Java, Indonesia. The fishing seasons of grouper fish are between August until March, which the peak fishing seasons are between April until July. The potential of grouper fish around Madura reached 158.9 ton, with the optimum fishing activities carried out by the fishermen of 1,179.166 trips. Unfortunately, the exploitation during the fishing activities by the fishermen is relatively high, particularly in spawning areas. Hence, it harms the coral reef as the habitat of grouper fish if it's continuously occurred, if eventually impact on the population of grouper fish around Madura Island. According to the field report, the exploitation of a number of grouper species is significantly growing, since it's one of the primary marine commodities in Madura Island. Therefore, it requires to conduct field survey to inventory the grouper species living in the ocean around Madura Island. This inventory of each grouper species is required to further illustrate the current condition of population, which is related with the latest conservation status. This research will further serve as a basis of future research that is oriented to groupers conservation based on morphology and molecular approaches. This research was conducted by employing field survey design to inventory the species of grouper fish around Madura Island. Madura Island is geographically close to the Java Sea in the eastern and northern parts, close to Madura Strait in the southern and western part, and this research was conducted during May until October 2019. This survey was conducted in fish landing bases in Madura Island, which are located in 4 regencies, that is Bangkalan, Sampang, Pamekasan, and Sumeneb. The location of fish landing bases is listed in the following table. The determination of the conservation status of each species found referred to publication in the IUCN Red List and Fish-based online database. In general, groupers have more than 1,000 species, of which most of them are important for commercial needs and relatively widely studied. To identify major threats to the grouper, the IUCN Red List criteria were used on 163 species. Southeast Asia has a number of threatened species. Not to mention, a number of species have not yet been identified, and some of them, which are in the status of near threatened, occur several other regions, while 30% of all species are included in the data deficiency. Grouper research on Madura Island shows that there are 3 genera divided into 14 species, namely genus Plectropomus, genus Cephalopolis, and genus Epinephelus. Genus Plectropomus consisting of Plectropomus maculatus, and genus Cephalopolis consisting of Cephalopolis cyanostigma and Cephalopolis buenat. And then genus Epinephelus consisting of Epinephelus fasciatus, Epinephelus ongus, Epinephelus merah, Epinephelus coioides, Epinephelus blakeri, Epinephelus heniocus, Epinephelus sexfasciatus, Epinephelus koyanus, Epinephelus erythrurus, Epinephelus analogus, and Epinephelus areolatus. According to the IUCN Red List, the conservation status of most grouper species found is categorized as least concern or LC, except Epinephelus blakeri and Epinephelus koyanus. Conservation status of Epinephelus blakeri is least concern in near threatened, while Epinephelus koyanus is included in the threatened species category. Threatened species and near-threatened species is a conservation status aimed at species that might be in an endangered or near-endangered state. Least concern is a category of IUCN for species that have been evaluated but not included in any category. Although most grouper species are included in the least concern, if it's not taken into account contemplatively, they will be categorized into threatened without being noticed. Epinephrylus blackeri and Epinephrylus koyanus in this study are the two species with the highest level of conservation status. Accordingly, both should be paid attention while carrying out fishing to avoid overfishing that damage the population later. Complete information about grouper cannot only help the assessment of the IUCN red list, but also to help conservation and management of these types of grouper. New information, specifically about the grouper taxonomic position, is needed to refine previous information and underlie further research in the future. The speed of decline in the number of grouper species is very worrying. It's feared that the local population will soon become extinct within a few years. If conservation steps are not taken immediately, it will have an impact on normal spawning habits, genetic diversity will decrease, and global grouper population density will drop to a critical point where species recovery will be difficult. The easiest step is to conserve the ecosystem and all species in it from the pressure of fishing and habitat destruction. The summary of this research showed that there were 14 species from 3 genus of family Serranidae was found at Madura Island, East Java, Indonesia. Based on IUCN Red List status, most of grouper species conservation status are categorized in least concern, except Epinephrylus blackeri and Epinephrylus koyanus. Epinephrylus blackeri, which was categorized into least concern in near threatened, while Epinephrylus koyanus was categorized in threatened species. Because of morphological similarity, molecular approach are needed specifically for the grouper taxonomic position. It's needed to refine previous information and underlie further research in the future. Thanks for watching this presentation, and in the end I want to thanks to the Biology Department, Faculty of Mathematics and Natural Sciences, Universitas Indonesia for supporting this research as part of dissertation research. And I want to thank to the editors and reviewers whose knowledgeable suggestion and comment improved this paper. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mr. Abdul Basit.
Yes.
This is the only one that is not about the fish. Yeah. Anyone have question, Mr. Nurul? Maybe you are interested the fish that Mr. Abdul Basit research. Anyone? Okay, before the question from the center participant, I would like to ask about this group of fish. How important this group, and why you choose this group? Mr. Abdul Basit.
Okay. In general, grouper have more than 10,000 species, of which most of them are important for commercial needs and are relatively widely studied. But to identify major threats to the grouper in the IUCN Red List criteria, there are 193 species, and in Indonesia, the identification of the species of these groupers is low. The identification and the taxonomy status and the IUCN Red List, and there is that's data. It's important for the conservation. Okay, and the grouper is very high value. There is in export and import commodities of Indonesia.
What the local name of this fish?
The local name is kerapu.
Oh,
Yeah, in
it's,
Indonesia,
yeah.
we call it kerapu, but in local, very local name, or in Madura Island, the society, the civil society or fisheries call them kerapu kendeng, and all the kerapu kendeng or kerapu pinter or kerapu another that's not similar with the species. Not always, yeah, not always represented the species.
Yeah, this is a very delicious fish.
Yeah.
Kerapu. I know kerapu tikus. Yeah. Nah, why you choose Madura Island? Because you stay in Madura, you live in Madura, or any
No,
other?
I'm just stay in East Java province, and Madura Island is one of island in East Java that's need the attention because the apa because the economic
Economic value?
Yeah, economical value, most of them from fisheries.
In Madura.
Yeah, in
And
Madura.
yeah, from your research, there are species that have category status near threatened. Yeah.
Yeah.
Why?
Until now, there are no specific reason about how there's near threatened or threatened, but it's I think it's the
Maybe the growth is so low, or they are predator, or
yeah, can be by predator and the ecosystem, and because of the fisheries activity or overfishing, like that. But I think I will because this part of the all of the dissertation research. So in the end of this research, I must have a social research to get the social activity and how the fisheries get the fish.
I'm very sorry I didn't catch your reason why the molecular identification are needed to the next research. Maybe you can give explanation for that.
Okay. For this research, it's the for this preliminary research, I use the catalog book FAO by Randall and Hamstra, 1993, that the grouper of the word. Okay, grouper of the word. And then in the catalog book, there are very similarity between species. So there's maybe any confusion to detection the species of grouper. So for the research, especially for this research, not just morphology, not just using the morphological approach to get the status of taxonomy and then the status of conservation for this group or species, and the molecular approach must need it.
Yeah, okay. The morphological characteristic is not enough to uh, take this phase of this species in the taxonomical uh, what you call
Yes.
In the taxa, in the, yeah.
Yeah.
Okay. I'm very sorry because my my research is in microbiology, so the fish is interesting for me, but I have low information about this, about the taxa of the fish. Okay, thank you for
Okay, thank you very
Mr.
much.
Abdul Basit. There's no question, Mr. Nurul? Maybe you have a question?
Oh, Miss Sri, thank you.
Okay, this is the last presentation. I think it should be me to present. I but I cannot present today because some of the reason, as I told you before. So yes, Samuel, could you give the link for the registration? Yeah. Okay. Every participant, please fill the attendance form in this link. The link is on the chat room, and we give you 5 minutes to fill out the attendance form. Okay, participants, all have finished to fill the registration form. Finished? Thank you. Yeah, you can answer in the chat room also. Okay. Thank you, all participants, ladies and gentlemen. We finally come to the end part of this parallel session. And before I close this session, I would like to thank so much to all presenters for informative and interesting presentation, and to the participants, but not very active participation. Yeah. Hopefully, this seminar will be beneficial for everybody. And before we close, before leave this room, we have a photo session. Okay, we have a photo session. Please turn on your camera and give the best smile, give the best expression. Please turn on your camera. We will take the photo session. Yeah, this pandemic make us can do this presentation from our home. Okay, ready, Samuel? Okay, not all turn the camera. Please, Miss Adelia. Miss Adelia. Okay, Miss Aelia. Sorry, Miss Aelia. Mr. Shan. Mr. Kugan. Miss Ayla, Mr. Kugan. Miss Luozaini. Mr. Shan, Miss Jennifer. Please turn on the camera. Mr. Abdullah. Mr. Nurul Muhammad Prakoso. Mr. Abdul Basit. Yeah. Ready, Samuel?
Okay, in the count of 3. 1, 2, 3.
Once again.
Okay, hold on. I'll copy it first. Once more in the count of 3. 1, 2, 3.
Okay, thank you all. Hope you are in good health and good condition, and success in the research. Thank you for attendance in the SMIC 2020. And Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you.
My
Thank you.
pleasure. My pleasure.
Thank you.
Thank you.